Hello, students. So, today we have to discuss these effector functions of antibodies. That how these antibodies will work, how these antibodies will perform their function. So, the basic purpose of the antibodies is that they will coordinate and will inform other components of the immune system about the entry of that foreign substance or antigen or microbe or bacteria or virus. So, first, when this microbe will enter and selection or colonal selection will take place, and specific antibodies producing B cells will be selected against a microbe, then antibodies will be secreted by the plasma cells, and these antibodies will perform different functions. So first, here we can see these antibodies will bind with the microbe and will activate the complement system. Complement system is a system of in the soluble proteins in the serum, in the blood, which are in the inactive forms. And when the complement activation will take place, and complement activation will take place at the time when the microbe will enter and the antibodies will be there. Antibodies will bind with the microbe, and then they, with the help of this FC part, they will activate the complement system. So basically, all the effective functions of the antibodies will help with the help of, they will take place with the help of FC part. And we know in the antibody, there are two binding regions, and one is the FC part. So this is the FC part of the antibody, and these two are the FAB binding regions. So here we see that this complement activation will take place. And when complement activation will take place, it will either will make MAC, membrane activation complex, and it will bind and it will generate pores in the bacteria and will result in the lysis of the microbe. This lysis will lead in the death of the microbe. So the first is the membrane activation complex. And the second thing is that when this complement activation will take place, it will form C3b, and these C3b receptors are at the macrophages. And so the C3b will bind with its receptors at one end, and other end it will mark this microbe. So it will promote opsonization and then phagocytosis. When phagocytosis or eating of these microbes by these cells will take place, macrophages, and they will destroy this microbe or will kill this microbe. So, and the third one is that when this complement activation will take place, it will produce C3A and C5A, and C3A and C5A will lead to the inflammation. So inflammation will help in the execution of or activation of B cells and T cells. So when antibodies will be produced, it will activate complement system, and complement system will work in three ways. One is the with help of formation of membrane activation complex or MAC, which will help in the lysis of microbes, and then C3b will be produced, which will help to mark these microbes and will bind with its receptors on the macrophages and will help in the phagocytosis. And also there will be C3a and C5a, which will help in the inflammation process, which will help to strengthen the immunity. Other thing is these antibodies, they will help to neutralize these microbes and toxins. So toxins are the poisonous substances which will be secreted by these bacteria, and these toxins, with these toxins, antibodies will bind, and then it will neutralize those toxins. And also, if there are some viruses, it will help in their neutralization. Here, the other thing is that there are FC receptors on the macrophages. So the antibodies will bind at one hand with the microbe with help of FAB region, and with help of FC region, it will bind with the macrophage because there are receptors for the FC part, and then phagocytosis will take place. So it will promote phagocytosis, and this antibody-dependent cellular cytotoxicity will take place, that when these antibodies will bind with these pathogens with help of FC part, they will bind with the natural killer cells because there are receptors of FC part, and then activation of NK cells will take place. They will secrete some chemicals, some hydrolytic enzymes and lysozymes, and they will take this target cell and will kill that target cell. So here you can see that the FC part is a very important part, which is helping to perform the functions of the antibodies. So FAB part is just marking the microbe, and FC part is helping to activate inform the different components of the immune system and activate the different components of the immune system, and then it will help in the destruction of the microbe. Okay, thank you.